Anyway, back to tonight. Megan Miller is our speaker tonight. Thank you so much for coming. If you've ever been to Clear Spring Ranch in Migration and you've seen a little table full of gadgets and gizmos and birds stuffed in prescription bottles, that's probably Megan and her team. And they are amazing to come across because you learn so much. She's also a big fan of ice cream. There she is with the Josh and John's flavor of the month, Toffee Talon. Megan Miller has been a part of Colorado's birding community since her first Christmas bird count in 2006. She completed a bachelor's degree in wildlife biology in 2017 at Colorado State University, and then spent some time doing field work across the country. She's worked on projects studying birds in apple orchards, seabirds, black-throated blue warbler ecology, and migration banding stations. In February of 2026, that's soon, she'll complete her master's degree at Colorado State University, Pueblo on the impact of wildfire smoke on migrating birds. Megan received a grant from Aiken Audubon to help fund this research.
We can start at the end.
And start at the end?
Yeah.
And go backwards?
Yeah. The whole time backwards.
These students, they're so innovative. Thank you, Megan.
Yes. All right. Thank you everyone for having me. Can everyone hear me in the back? Yeah. All right. If you can't hear me, raise your hand, shout my name, etc., and get my attention. All right. I am Megan Miller. I am finishing up my degree at Colorado State University, Pueblo, in the lab of Dr. Claire Ramos. And I am evaluating the effects of wildfire smoke on the oxidative status of migrating birds. That's a very big title, and we're going to break it down piece by piece so it doesn't sound so scary, because I was very scared when I first started this project. All right, we're going to start on like a very big scale and kind of work to a much, much smaller scale. So in the past 20 to 50 years, there's been major shifts in the wildfire regime, not just in the United States and Canada, but all around the world. So as many people who have grown up and lived in Colorado their whole lives know, the fire season is becoming longer and longer. Habitats that don't see a lot of fire are seeing more fire, and habitats that see a lot of fire are seeing less fire. So it's changing how the succession of these habitats turns over, and that changes how these habitats are structured fundamentally. And then in addition to that, it's increasing human smoke exposure across the board. And I wanted to include this really interesting graphic, but I couldn't find it. But globally, and in the United States especially, there's been a decrease in poor air quality. So air quality has been getting better in the last 20 years, but in the last 5 years since COVID, wildfire smoke has kind of reversed that progress, because the fires have been so severe that they've been able to kind of offset all of that progress that we've made. And humans have the opportunity to take shelter indoors. We have fancy air purifiers, we have HVAC systems, but wildlife are exposed to this fire and the smoke 24 hours a day, and in some cases year round, depending on the location of the wildfire and the individual bird. So we're going to talk about particulate matter a lot, and there's different sizes of particulate matter. And the one I'm focusing on is PM 2.5, which is particulate matter 2.5. And that is like tiny particles that are smaller than 2.5 microns. So that's smaller than beach sand, smaller than a hair. And these blue dots here represent dust or pollen or mold. So it's even smaller than those. And this particulate matter can be made up of organic compounds and metals. So that's things like mercury. Oh my gosh. Well, selenium is one that gets kind of moved through the atmosphere when like wildfires burn like industrial areas. It has impacts on kind of two major systems in the body, but once those are impacted, they can kind of spread throughout the body. So the pulmonary and cardiovascular system are.
I never shared the screen, so the people on Zoom
are
being they're being so polite right now. But one of them is a mole for me, and they just told me that I'm a silly goose. Oh, there's the big share button. Oh my goodness. Hey, people on Zoom, somebody unmute yourself. Can you see the screen?
Yes, we can.
No worries, no worries. All right, so you can see this diagram here of the different sizes of particulate matter. We have the fine beach sand, the hair, the dust and pollen, and then finally the little red beads is PM 2.5, the smallest particulate matter. And when you breathe this in, it goes into your lungs. And in your lungs, it can be transferred to your cardiovascular system. And from there, it gets spread throughout the entire body, and it can impact like neurons and neural firing. It impacts the health of your lungs, obviously, and it can increase cardiac issues amongst humans and almost certainly birds as well. So why is smoke bad? Other than what I just said with the physiologic impacts, smoke moves. And in the 2020 wildfires in Australia, there was a university that did some smoke modeling, and they followed this like plume of smoke as it like moved across the globe, and it persisted in the atmosphere for over 6 months, and it moved, I think 60,000 kilometers, 6,000 kilometers, a very long way. It moved to a different continent, basically. So, you know, it doesn't just impact where the fire is starting, it impacts all of the land as it like moves around the globe. And then, of course, birds move. So here in the United States and Canada, we have our high boreal forest where we're seeing a lot of wildfire and a lot of burning in the recent years. And I think Canada in 2025 had its biggest wildfire year of all time. So that means that all of our neotropic birds that are breeding up in the Arctic, up in the boreal forest, are being exposed to this smoke in their breeding ground. And then during migration, during the wildfire season in the United States and the West, especially, you know, the Rockies through California, they're experiencing wildfire smoke during migration. And then also in Central and South America, places like the Pantanal in Brazil, and other places in Central and South America are seeing, again, major wildfires and these mega fires that are bigger than many of the historic fires of the area. So that's fires in all three major areas of this bird's life history. And there's kind of two main ways that smoke can impact birds and bird health, and that's through their behavior or their physiology. So I'm just going to do one slide on behavior because I think it's interesting, but it's separate from my research. This is a really interesting study that they did on some greater white-fronted geese. So they had these geese GPS tagged on their breeding ground, and on the left side is the 2019 route of the geese. So the blue line is the goose's route as it flies south, and the little circles are where they're stopping to refuel for migration. So they're flying, 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 they stop, they eat a bunch of food, and then they fly some more. So now on the right-hand side, all of that red is smoke in the atmosphere. And you can see how much the geese moved around that smoke to avoid having to fly through it. And that extra distance totaled over 270 kilometers per goose. And I think the caloric offset was equal to 2 to 3 days worth of forage. And as you can imagine, if you're this goose and you're all the way over here in Idaho, if you know what that area looks like, it's all kind of like high sagebrush steppe. So if you're a greater white-fronted goose and you're used to foraging on the coast of Oregon and California along these rich wetlands, and you end up in the high steppe of Idaho, you're not going to be able to refuel to continue your migration. So we don't really know how this kind of forced dispersal is impacting birds and their migration paths. So this was just kind of like a lucky happenstance that these geese were tagged. They were not tagged for this specifically. It just kind of happened that way, and they got some great data from it. So the other side of this is the physiology. So when birds breathe in these fine particulate matter, there's all sorts of things that can be impacted in their lives. So the 2025 Krieg paper came out very recently. And this is a cool experiment where a researcher has had house wren nest boxes, and they've been doing just like nest surveys of these nest boxes for several years, so they can compare years with low air quality or good air quality to years with bad air quality. And the years with bad air quality had depressed nestling growth. So that means the nestlings, they grew slower, they had lower time to maturation, and then lower fledgling success. And then our pigeon over here, there's a fair number of research, or a fair amount of research on like industrial air pollution from like factories and stuff like that. So this pigeon paper is really interesting because they took some pigeons from different areas of the countryside in Europe, some with a lot of pollution and some with less pollution, and they found that birds living near areas with a lot of pollution had immune suppression. So they were finding that they weren't able to fight off infections. Their bodies were unable to kind of help them survive in the long term because they're unable to fight off those infections. All right, so this is kind of a big jump into what I focused on for my project, which is oxidative status. So there's two sides to oxidative status. And when you hear about cranberry juice having antioxidants that battles free radicals, that's actually a real thing. It's not just to sell cranberry juice. So reactive oxygen species are created during the metabolic pathways and process in your body. So when you exercise, when you're sleeping, when you're eating, all of that creation of calories, creation of energy in your body creates these free radicals. And it's normal, except in certain circumstances. So these free radicals are highly volatile, and that's good if you want to create energy in your body, but bad if there's too many of them bouncing around. And because they're so volatile, they can grab onto different parts of the cell and they can destabilize those parts of the cell. So that might be DNA, that might be RNA, or that might be the cell wall. So your body has antioxidants that can come on and attach to those cells or to those molecules, and it will stabilize them. So one of the things that can cause an increase in reactive oxygen species is exposure to pollutants. And a lot of, whether that's through air or water or food. So where do they come? I already talked about all of this. There we go. All right, so why are reactive oxygen species bad? So there's a lot of research on reactive oxygen species because they're kind of this like interesting thing that people don't know that much about. Birds with high oxidative damage or high reactive oxygen species or free radicals, all the same thing, they had reduced fecundity. So that means that they laid fewer eggs. So females with high oxidative damage laid fewer eggs. In the house sparrow, total antioxidant capacity was lower in urban areas. So they suspected that it was due to pollution in the bird's body. Birds that lived in urban areas had low antioxidant capacity, so they could not combat the free radicals, and birds in less polluted areas could combat free radicals because they had high antioxidant capacity. And then in Scopoli shearwater, birds with high oxidative damage had reduced fledgling success. So they might have hatched a normal number of young, but the number of young that fledged was lower. All right, so antioxidants, there's two general types, and we're not going to get too in the weeds for this, but just know there's ones that are created inside the body, and then there's ones that come through the diet. And you don't have to memorize any of that. But I do just want to point out this diagram. The antioxidant is on the left, the green one, and the free radical is on the right. So basically what the antioxidant does is it just donates an electron to the free radical, so then it is a stable element. So because the free radical doesn't have an electron there, it really wants to pull an electron from something else. And when the antioxidant quenches that electron, it no longer wants to grab onto things. And Lacey helps me make that. Okay, so I think one of the hard things about this research is different tissues have different metabolic activity. So something that might be bad for heart muscles might not be bad for leg muscles. So it gets kind of complicated when you're trying to decide which tissue you want to look at. And I think that's interesting here because this is a Eurasian sparrow in China. And in the polluted areas, they had lower antioxidant capacity in the heart and lung. But over here in the in the polluted areas, they had higher antioxidant capacity, which is confusing when you're trying to create an experiment for this. And then what is that called? White stork. So they had reduced circulating antioxidant capacity living in polluted areas. So chicks that were growing up in polluted areas did not have antioxidants, or they had lower antioxidants. So you want an ideal balance. If you have a lot of antioxidants, you're going to have lower free radicals or lower oxidative damage. And if you have too many free radicals, you're going to have depleted antioxidant capacity because it's kind of being absorbed by all these free radicals. And then if there's not enough to quench them all, you're going to increase your oxidative damage. And that could be, again, damage to DNA, damage to the cell wall, damage to organelles in the cell. All right, so my major hypothesis is that oxidative status markers, which is reactive oxygen species and antioxidant capacity, are influenced by exposure to wildfire smoke and body condition. So my predictions are, as pollution increases, DNA damage will also increase, because that's what I measured was DNA damage, and I'll talk more about that in a bit. And there's some research that shows that heavy metals increase the production of ROS. So those heavy metals that are present in the fine particulate matter of wildfire smoke are going to move through the circulatory system, potentially increase those reactive oxygen species. Then as pollution increases, antioxidants will decrease because the antioxidants are being used up to process all of those reactive oxygen species. And then as emaciation increases, I think antioxidants will also increase. And that's because the catabolism of protein, or when the body breaks down protein or muscle tissue, it creates uric acid, which is a very strong antioxidant. And if you're a bird and you're migrating thousands and thousands of miles, you're going to be breaking down muscle tissue, and that's going to turn into uric acid, which is an antioxidant. All right, now for the fun part. So this is all the field work we did. I did, all of my data came from the year of 2025. So even though I've been at Clear Spring Ranch for a couple of years, I only included 2025 in this experiment. So we were in the field during migration, so April through May and August through October, 5 hours a day, 6 days a week. And I was not there all 6 days a week. I was there, we split it half and half, my advisor and I. And then here's the breakdown of the blood samples. So this is not all the birds we caught. If you've been to the station on a busy day, you know we've caught way more birds than this. But I took blood samples from each of the birds that we caught. So this is just the number of samples that I had enough blood from that I was able to process for my research. And I think everyone here knows that Wilson's warbler is probably the most ubiquitous species in fall migration. So I have a lot of samples from them. Oh, there we go. Okay. So our air quality, we have an air quality monitoring station at Clear Spring Ranch, which gives us 24-hour readings of the air quality. So in theory, the moment I'm banding the bird, I can get the air quality, which is a little bit too fine a scale for what I need, but it's kind of cool that I can actually do that. And unfortunately, the year that I did my research, we did not really have any major wildfires, but of course I cannot wish for wildfires, so I am both happy and disappointed. But so this is what I have circled here. This is the air quality index that the APA puts out. And for our year of data collection, we only got up to a moderate level of air quality, which is great, both good and bad. Bad for research, good for people, good for us who had to be out there every day. We didn't really want to be out in the poor air quality, but so we didn't really have severe air quality, but it did get up to the moderate scale. So that's moderate for people with asthma or other pulmonary issues. And then I just had a reminder of the particulate matter scale. So yeah, we're going to keep that in mind going forward, that all of the results don't necessarily stay the same as you get more severe air quality. And as I said, so we had 16 days of moderate air quality, but the air quality machine only read about 4 days of moderate air quality because just the way air moves around. Some days were higher than others. Some days the smoke would be pushed out to the plains or closer to the mountains. So we did a lot of sampling on those 4 days so I could get as much data as possible. So this chart shows the smoke days, how many birds of which species we captured, and then the year, how many birds I sampled. So you can kind of get an idea of the proportions. So like our white-crowned sparrows, out of those 40 total samples for the year, 20 of them were sampled on smoke days. And this photo was not taken at Clear Spring Ranch, but I thought it was a great photo of wildfire smoke with a bird. This was taken during a controlled burn in Victoria, Canada, where I worked this past fall. All right, so the lab work. So I took blood samples from as many birds as possible during our smoke days. And blood sampling is kind of weird if you're kind of woozy around blood. Don't think about it too much. But we just give them a little poke right in a vein in their wing, and the blood pools. And then we use this thing called a capillary tube. And it's not a syringe or anything like that. It's just a glass tube. And when you put it up to the drop of blood, the capillary action sucks the blood into the tube. It's very cool. And then I take that back to the lab on campus and I centrifuge it. And when you centrifuge it, it separates the plasma from the red blood cells because the red blood cells are more dense. And that's what this picture is over here. So you can see those densely packed blood cells at the bottom and then the yellow plasma up top. And I think that picture is interesting, but it's a side note. The whiter one, the bird has more fat in its plasma. So it just has like more circulating fat, which is why it's white rather than clear. I just thought that was really interesting. So I take the plasma off the top and then I freeze it in a very fancy -80 freezer. So then from each one of those plasma samples, I separate it into 2. So I'm going to do 2 different assays. And an assay is just a way to analyze the blood samples. So like when you go and you get your cholesterol test or you go and get your blood sugar tested, it's going to run it through an assay, or some lab tech is going to run it through an assay. So we're doing that for birds basically. So the 2 assays I'm looking at is a total antioxidant assay, and that is going to help us determine what the circulating antioxidant is. So like I said, it's different in different tissues. So we're looking to see what is circulating in the blood. So it's not going to tell us what's in the breast tissue, it's not going to tell us what's in the heart. So it can't kind of give us insight into that, but it'll help us understand like what is being circulated in the body actively. And then the other assay we did, I don't know if I can say this more than once, so I'm just going to call it 8-ODOG, is what me and my advisor call it. And that measures the damage to the DNA. So like I was saying, these reactive oxygen species, they cause damage to DNA. And when that damage is caused, this 8-ODOG is produced specifically due to oxidative damage. So we can measure the concentration of that and know that that is the damage done specifically by these reactive oxygen species. And both these assays took a lot of time to learn how to do, but I did read about 10 books while I was doing all of them. I listened to about 10 books while I was doing all of my lab work, which was great. Okay. So, all right. So in addition to having those 2 assays and the values that I got from that, these are the other condition variables that I wanted to look at. So we're going to look at PM 2.5. Obviously, that's our pollution variable. We want to know the species. Are some species more impacted than other species? Long distance migrants, short distance migrants, frugivores, insectivores, etc. We wanted to look at season. In theory, birds in the fall could have higher antioxidant capacity because there's more fruit available. More fruit means higher antioxidants, etc. We wanted to look at season. We wanted to look at fat because we collect fat on all birds. It's easy to measure. It impacts their metabolism. Fat holds a lot of antioxidants. A lot of the antioxidants are fat soluble. And that's a whole nother rabbit hole, so ask me questions about that later. And then muscle. And I think muscle is really interesting. It's not a score that I take very often at different banding stations. And Claire takes it at hers, and I think that's really interesting because I think there's a lot of opportunities to do interesting things with it. So when you think of the chest of a bird, they have a big keel, kind of like the keel of a ship, and their muscles are very rounded across that keel. And when birds catabolize those muscles, when they use those muscles as fuel, the muscle kind of atrophies a little bit and the keel protrudes. And this little green device. It's just 3D printed. You can kind of hold that against the keel and it measures that displacement of the keel from the muscle tissue around it. So a bird that is emaciated has a higher score, and a bird that is not emaciated is going to be closer to 0. Okay, I'm going to do the stats, but don't panic. I'm going to do my best. So I added some extra stats slides for practice for my defense. But so let me know if anyone understands this. Okay, so I have 2 sets of models, right? So I have my antioxidant capacity and I have my oxidative damage. And then in the previous slide, I had all those different variables that I'm going to be comparing against each of those. So these are all different model types, right? So I'm combining these models against each of these variables to determine which model or which combination of variables might be impacting one of these 2 in a positive or negative way, right? So I have my top model here, which is pollution, season, fat, muscle species. That's everything. That's my global model. And then we did a variety of different combinations of different variables to see which ones were the most likely to be influencing these 2 different other variables. And I think there's like 30 some models total, and I didn't want to put them all here, but if you were very curious about them, I will show you. All right. So I used something called AIC, or Akaike's information criteria. And the way that works is it takes all of those models that I showed you in the previous slide and it ranks them as most likely to explain variation in antioxidants or oxidative damage. So here is my antioxidant table. And from the top to the bottom, it shows most important to least important. And you can see my null model came out on top. So what that means for me is that there is no measurable impact of any of those other variables on antioxidant capacity. But it gets more complicated, as it always does. So we're going to say what's important here. That's the delta value. So over here, you see AICC, delta, and weight. So your top model is always going to be a delta of 0. 0 to 2 is where you want it to be. So we see our top model is 0. And then the weight. So models with similar weights are less distinguishable. So over here on the far right, we have weight. And you can see those top 3 models, the weight is extremely similar. They're almost the same. So that means those top 3 models, you can rearrange them in any order and there's no support for one over the other. So still doesn't really help me, right? So our null model says there's not much information in our different variables, and that is supported by the weight as well. But we can also say, are there any repeating variables? So variables that show up in multiple top models are more likely to explain more variation. So our repeating variables here are season and muscle, which I think is interesting because, as we talked about before, birds that are catabolizing muscle have higher antioxidants because of the uric acid produced during the catabolizing of muscle. And then season, we talked about how different seasons have different dietary antioxidants available, so that might also be impacting. But because there is so much uncertainty in these models, all I can say is maybe. You can ask me again in like 5 years, and we'll see if anything else is important. So this is just a reiteration of what I said. We have the colors, the null, muscle, and season are similar weights. Season and PM 2.5 are lower weighted models. Yes, there's little support for any model over any other model. All right, and then I made some graphs just to give you all some relief. If there was a relationship between these 2 variables, there would be a slope on these lines. But since they're basically flat, that means, again, very little support for any of the variables that I found. All right, so next is our oxidative damage table. So again, same thing. We're looking at the top models are between 0 and 2. So species came out as our top model for this one. And if we're over here on the weight is quite different from the one below it. So we can say somewhat confidently that there's more variation in oxidative damage between species than there was for any other variable. So season, PM 2.5, fat, and muscle had very little impact on oxidative damage, but species had a high impact on oxidative damage. Yeah. All right, and here's my cool table for that. So this is mean oxidative damage for each species. So I have my species down here. Sorry, I used alpha codes. It's a lot easier to fit those than the full name. And the letters that are most similar are not statistically different, right? So A and B are not statistically different from AB. B is not statistically different from A and B, but B is statistically different from the A's. So we can say that the dark-eyed junco has the highest mean oxidative damage out of all of the species that we tested, and it is significantly different from all of these species that have lower oxidative damage, but it is not statistically significant from these in-betweeners that are A and B. And this could be interesting for a variety of reasons. And I did a couple other analyses breaking it down by family to see if there were different families impacted differently. And I didn't really find a ton of patterns. But the thing that I think is most interesting is most of the juncos we caught were on the smoke days. And they did show up as the bird with the highest oxidative damage. And statistically, it came out as insignificant, so I can't say that. But it is something that if I had another 2 years, I would be testing every junco we caught because I'm very curious. But I'm also curious to know more about this layout. You know, there's not a lot of research done on oxidative damage on birds. I think when I was doing research for this assay, I found 2 publications that had used this assay on birds before, and mostly it's done on humans. So I think there's still a lot of information to be had here. And if I had another 2 years, I would do it. All right, so here's our graph showing no relationship between oxidative damage and PM 2.5. So my hypothesis, which I was very excited about, says that there's no relationship. But again, it can't really be said that there isn't any severe damage because we had such low air quality or good air quality, right? So it only went up to moderate. So, you know, our like really terrible days when we had like ash falling from the sky, it really doesn't account for days like that. Okay. All right, so my main takeaways. Muscle and season might be important for antioxidant capacity. Oxidative damage varies the most within species rather than because of pollution or muscle or fat. And low wildfire smoke concentrations limited our ability to measure the influence of particulate matter on oxidative status. And then there's a lot of uncertainty in understanding how the reactive oxygen species and oxidative status functions in birds, just because it's a hard thing to measure. It's a hard thing to get repetitions of. You can only take so much blood from a bird that weighs 10 grams, and then to catch it again next year and take blood again, it's almost impossible unless you're working on the breeding grounds. So this is kind of like a very new area of research in terms of like wildfire smoke and passerines. So I'm excited to see where it goes from here. There's, like I talked about earlier, there's the study showing depressed nestling growth. There's a woman, Olivia Sanderfoot, who has done a ton of wildfire smoke research, and she's super interesting. She did an interesting project showing that smoke events alter the dawn soundscape. So I think that's really interesting because that also falls under our like behavioral issues, where like birds are not only being impacted at like physiological level, it's impacting like their potential breeding success through their like lack of dawn chorus. And then, this is another, so Sanderfoot is a co-author of this other paper too, Wildfire Smoke Impacts the Body Condition and Capture Rate of Birds in California. That paper kind of, what did they do? They did number of days of severe smoke previous to the date of capture, and birds that were captured after like a week of severe smoke had lower body condition than birds that were captured after a week of great air quality, right? So there's like some research showing that wildfire smoke is causing health issues in birds, but it's very hard to measure because oxidative stress is difficult to measure and understand. There's more research conducted on industrial air pollutions and in lab settings because it's easier to control where the smoke is, where the pollution is, where the birds are. And then of course it's difficult to mobilize and sample safely during wildfires because you don't really know where they're going to show up, and you don't necessarily always have a field crew that can don respirators and all these like safety equipment to run out and take blood samples from birds. It can be kind of a challenging thing to do. So there's many things in the way of future research, but like we're all stubborn enough to make it happen. So. Okay. Thank you very much. I have so many people to thank for this research. My advisor and my committee members, the Ramos Lab undergrads who came out into the field with me and did all of this without me having to beg them, which was wonderful. Then my friends and family, Jordan and Lacey and Kelsey are here. And then all of the people who have helped me get through this. Kyle is constantly helping with my statistics. Viridiana came out with me on so many cold mornings when no one else was signed up. And then of course, my family. So thank you to Aiken because those assays were very expensive and Aiken paid for most of them. So that was very nice of them. Anybody have any questions?
Ask one. I'm Paula. With the grant money, did you use it to pay for the assays, or was it for the whole thing, or?
Yeah, mostly for the
how did it help
assays.
you, I guess?
Yeah, so the assays, so each assay can test about 30 samples, and I tested 550 samples, I think, and each assay costs about $400. And I have to test each sample in duplicate for statistical reasons, so that really limits the number of assays or tests I can get per assay. So I end up having to buy, I don't know, it's like 15 to 20 assays total, which ended up being a lot of money. So the lab equipment was very expensive for this project, and that's how I spent the Aiken grant.
Forgive me if this is a dumb
No,
question.
never.
How do you account for the fact that each of the individual birds would have different exposure? One might be very exposed, one might be lightly exposed,
Yes.
in the same area.
That
One might have flown right through the wildfire, and that one just was on the periphery.
Yeah, that was one of the biggest things that we discussed early in our discussions of how we were going to plan this research project. And I think no one has really been able to answer that yet. Because in order to know that, you have to like have a tag on the bird, isotope maybe analysis, but then you're adding another $5,000 to $10,000 of lab work if you want to do isotope analysis. So I think that's why we decided to do the air quality at the field site, so we could do like the date of capture to try and get it. And also I do have subspecies for some of these birds. So in theory, I could say, well, you know, slate colored juncos are coming from here, so it's unlikely that they're going to hit the wildfire over here. Or like the white crowned sparrows, you know, we have 2 subspecies of white crowned sparrows, but I haven't looked at that yet because I haven't had time. But that's kind of one of the big questions with wildfire smoke right now is how are we going to decide where these birds are coming from and how are we going to measure that? So if you have any ideas, let me know.
I was impressed you were able to do what you have done.
Thank you.
So when you're graduating next month, is that what it is? Do you plan on maybe like trying some more research in this field, or is there another field that you are interested in kind of researching and, you know?
Yeah.
What are you interested in?
Yeah, I definitely want to stay in the wildfire realm, because I think there's not a lot of research relating to smoke specifically. So I'd like to stay there if I can find a lab that does smoke ecology. Because I think there's many more questions to answer and many more fires will be had, I think. Yeah.
Well, when you need a grant, you know, one organization to come to.
Definitely, yes.
Thank you.
Thank you.
Yes, thank you.
Hey, when does banding start at Clear Spring this spring?
You know, I have no idea. I should give a shout out to Kelsey, who's in the Zoom. Kelsey is now running the banding station. Lacey, you might also know, is Kelsey's second in command.
Yeah, what was the question?
When does banding start this?
Oh, I think we're probably gonna decide that on Friday.
Okay.
So-
Yeah, we're having our first lab meeting this Friday, so. Usually we start like third week of April. I think that's when we started last year.
Sounds right.
Yeah.
We do have at least one more question here,
okay.
in the chat. How does the size of the migrating birds affect their susceptibility to pollution? Warblers and cranes are pretty different.
Yeah.
I would agree. Would the bigger bird be more resistant to pollution?
Yeah.
Was there any? I remember that chart where like juncos, and then it was like.
Yeah.
Pelvis, was it?
That's a great question.
But I don't remember what was in the middle.
Yeah, let me, is my PowerPoint still up? I think, I have another slide at the very end that has the family breakdown. Let me see. So this is the, ooh, let's make it bigger. Okay, so this is the family breakdown I did just out of curiosity. And it looks like, I mean, warblers are kind of on the low side, but vireos are on the high side. And they're roughly the same size, a couple grams off. Yeah. And troglodytes are wrens, so they have higher oxidative damage. Yeah, I don't know what icterids those are. Those might be orioles. But I think we didn't catch that many orioles. So it might not be a good representation. And the cardinalidae, that's just western tanagers, I think. Yeah, that's a great question. I have no idea. I'll have to see, sort it by weight or a mass and see if there's any interesting insights. Yeah, no answer to that question. Ask me again in 6 months.
PhD.
Yeah, exactly.
Hey, that's all we got.
Pretty great.
Thank you, Megan. Keep an eye on our calendar whenever we do award a grant. We typically ask the awardee to, if not a full-blown program that Megan presented tonight, if at least it could be like a 10-minute introduction on another night. So we usually do ask those awardees to give a little bit of a summary or a big summary to us one evening. So just keep an eye on our events, and maybe there will be another one coming up.
Hey, Jessica Talley also asked a question in the chat. Sorry, voice from the clouds.
Thank you, because I can't see the chat. Oh, I like that. What was the most enjoyable part of your research?
Gosh. I mean, yeah, catching birds is great. I love catching birds. Like the first month of the season is great. The second month is great. The third month, I'm ready to never look at a bird again in my life. But then like 2 months later, I'm like ready to touch birds again. So I like that. And I really liked the lab work a lot. I think it was like very peaceful to just sit in the lab all day, and I listened to my audiobook and I pipette for 8 hours, and it was really nice after like all the hard field work to just like relax. Yeah. Yeah. Yeah.
I do have a question, one more question. So you said I asked you in 6 months, will you continue to study after
Yeah,
this, after you're done?
Definitely, yeah. I haven't decided where I'm going yet. Interviewing lots of places. But I definitely intend on continuing this research in some capacity. And even, I don't really know what else is going on out there in terms of like research. So like someone else could figure it out before me in the next 6 months. So I think you never really know like who will publish what.
Okay.
So yeah.
That's exciting.